Yes, go ahead. Thank you.
Yes. Okay. Very good evening to all respected academicians, clinicians, researchers, and passionate learners who joining us today. I am senior dietitian Souvik Chakraborty. I'm this PhD scholar in nutrition, CD. I've done my MSc and B Pharma, PGDP, CNA, and certified sports nutritionist as well. And I'm an executive member of Community Nutrition Core Group. So, on behalf of IPIN India Association and the Community Nutrition Core Group, IPIN India, it is an honor to welcome you all to this thought-provoking session on precision nutrition and bridging community health and genes. So, we are living in an era where nutrition is no longer just about food on plate, but about information encroached within our gene. So, precision nutrition empower us to move from reactive healthcare to proactive and preventive and personalized nutrition, transforming the way we serve individual and communities alike. For the country as diverse in India, this approach is not a luxury, but this is nowadays is a necessary. So we are privileged to learn from today from Dr. Janani Tamilavam ma'am. So let me introduce ma'am. Ma'am is a director and Institute of Nutrigenomics, and she has done Bachelor in Food Science and Nutrition and Food Quality Control. And ma'am has done Masters in Food and Nutrition from PSG College and Arts and Science and Erasmus Munds, the Masters in Food Science, Technology, and Nutrition from Berlin, and PhD in Food Science and Nutrition, specialist in precision nutrition and obese. And ma'am is professionally a director in Institute of Nutrigenomics from 2017 and founder of Nutrigenomics Specialist and scholar nutrigenomics, nutrition consultant and research scholar in product in university in USA, and senior dietitian in Village VLCC, Middle East, Dubai, and DHA certified and nutrition expert. Ma'am's research publication is multiple peer review publications on gut microbiome and obesity, nutrigenomics, and nutritionally tailored diets, and genetic testing and personalized nutrition in India. Ma'am, her journey remains us when science meet compassion and nutrition become tool of lifelong health. I am confident this session will ignite new ideas and inspire innovation and shape the future of nutrition practice for all of us. With a great respect and enthusiasm, I invite Dr. Janani ma'am to begin. Ma'am, it's your session.
Thank you, Souvik, for the nice introduction. So I'll just directly move
Thank you,
on
ma'am.
to the session. I'll be screening the PPT now. Let me know if it is visible.
So you can share the screen now.
Yes, it is perfectly visible.
Perfect.
I hope this is visible, right?
Yes.
Yeah. So good
Perfect.
evening, everyone. Thanks for the invitation. Today we'll have a short session on precision nutrition, and which is going to bridge the community health and the genes. We all know that one size doesn't fit all, and that is what nutritional genomics is all about. So it's about personalizing one's dietary needs based on the genetic makeup. And thanks for the opportunity for the webinar that's organized by the Community Nutrition Core Group, IP in India. You're doing a wonderful job of educating the nurtured ones, as well as the seasoned professionals are also getting kind of upgraded with the knowledge that you are transcending. So congratulations to that, and thanks for inviting me again. So moving on to personalized nutrition. So first, let's learn about the terminology first and what is the way forward. So if you look at stratified or tailored nutrition, so this is the approach that has been followed for decades together, where we assume that a group of individuals with shared or similar characteristics to have been suited for a similar intervention. So for example, our RDA values, which are kind of consolidated by the National Institute of Nutrition or for any other authorities in any other countries across the world. So they all come up with population-based dietary guidelines, so which is stratified or tailored nutrition. So the nutrition is for a group of population, and that segment of population will have similar characteristics like the occupation, the activity levels, the gender, and so on. And then what we found out was there were some missing links or gaps that existed in the stratified or tailored nutrition approach. And we thought about like personalizing it. And personalization is a very broad term. So if you're looking at preferences, North Indian, South Indian, Eastern Indian, or any region-specific preferences, when you take into account, that is also called as personalized nutrition. But what we are talking about personalized nutrition is we're also getting in depth at elemental level, where we look at the individuals in a systems biology approach, and we look at only one component or like a single layer of the person apart from the normal dietary assessment that we make. Whereas precision nutrition is the way forward, where we get a sufficient quantitative understanding about various relationships that a person can have and what is the interaction between his or her phenotype with that particular different components that we're talking about. So this is like moving ahead from a population-based dietary guidelines approach or one-size-fits-all approach to more of personalized, and to move forward, it would be more precise and accurate in nature, which we call it as precision nutrition approach. So you could see that the transition happening, and rather than looking at the whole population, we are moving forward to looking at an individual, and in the way forward in the future, we'll be looking at different complexities of each individual. So you can assume that there are different layers among the individual which we are going to look at. So this is the kind of where we are moving ahead to a picture. So as I told you, if you have a population-based dietary guidelines, so it would have the information about what are the different food groups, what should be the serving quantity, and it would be well balanced. There is no doubt about it. We need to have such a balanced diet, but this may not consider the individual differences that exist among individuals. Like if you could look at the picture on your right hand side, so it has different pictures of different age groups, different gender, as well as different ethnicities. So we don't really look into the differences that exist within the individual and differences that exist among the individual of the same population. So this difference is what is contributing to the individuality and the human metabolic individuality, we call it as, and this is the difference between one size fits all approach and personalized nutrition approach. You no longer look at individual as a part of the population, but rather than an individual with its own uniqueness, which the nutrition plan has to cater to. So this is the difference. So if you look at conventional meal plate, a thali that we usually have in India, so we actually consider the ABCDs of nutritional assessment, which is anthropometry, biomedical, clinical signs and symptoms, and dietary assessment. So if you look at the ABCDs of dietary assessment, so this is like you are just looking at the phenotypic assessment or a symptom-based assessment. When I say phenotype, it refers to the descriptive characteristics of an individual. So let's say whatever we are able to observe, whatever is visible, whatever we are able to measure, like the biomedical parameters, or whatever we are able to see and measure from a person, it's called as a phenotype. So far, the conventional nutrition approach has been relying on the phenotypic measurements or the phenotypic assessment. But when you look at the personalized meal plate based on the genes, it could be many elements of personalization, as I said, because personalized is a term that is a very broad in nature and it has a lot of elements to it. So when it comes to anthropometry, biomedical, clinical science, and dietary preferences, how are you going to integrate the genetic factors is the additional personalization element that is going to be added to the conventional nutritional assessment. So phenotype, as I said, whatever the descriptive characteristics that exist within a person, it refers to as the clinical phenotype, and dietary intake or physical activity, environmental factors. So all these we have been considering in the past or in the conventional nutrition assessment. Whereas when it comes to omics integration, these are like, again, genomics is very vast like nutrition. So when we talk about genomics, it's only a tiny bit of the whole genome that we are talking about. Whereas there is proteomics, transcriptomics, metabolomics. So all this, when it gets integrated with the phenotypic assessment, we call it as precision nutrition. Talking about in brief about genomics. So when we talk about the DNA and the information that is carried in the genes, it refers to as genomics, and we study about how nutrition interacts with the genome. It's called as nutritional genomics. And ultimately, genes carry instruction to make a protein, and collectively proteins, the mass is called as the proteome. And when we study about how nutrition interacts with proteome, it is called as proteomics. And transcriptome, you all know that food acts as a signal and gives the instruction to the cell what it has to do. So you need some transcription factors for this to happen, for your food to turn into signal and tell the cells what it needs to do. So the collection of all the transcription factors, the messenger RNA, together it's called as transcriptome. And when we study about how nutrition affects transcriptome, it is called as nutritional transcriptomics. And you all know whatever you eat, whether it's roti or rice or fish or meat, there are several metabolites that are formed in the process, whether it's carbs, proteins, fats, whatever you eat. There are numerous metabolites that are formed within our body as a result of metabolic processes. And collectively, all the metabolites put together forms the metabolome. And when you study about how nutrition and metabolome interacts, it's called as nutritional metabolomics. So when you integrate all these omics, genomics, proteomics, transcriptomics, metabolomics to the phenotypic assessment of nutrition, then we call it as precision diagnostic nutrition assessment, where you get a very individualized, specific treatment plan. So this actually is, we talk it as phenotype and genotype correlation, or when you combine phenotype and genotype, it is referred to as precision nutrition. So this is what precision nutrition, the term and science, are all about. So nutritional genomics, as I said, it's a very tiny part, but we are going to learn about the tiny part in today's session. As I just to show you this picture, it's quite a short time to explain, but I'll try my best to convey the message here. So if you look at the DNA, so you have chromosomes. When you look into the cells, you have the nucleus, and within the nucleus, you will find this chromosome. And within the chromosome, when you untangle it, you will find something like this, which is called as a DNA. And DNA, you have the genes, the segment of DNA are called as the genes, and you will have these base pairs, adenine, guanine, cytosine, and thymine. And this DNA, as I said, it forms the basis of nutritional genomics. So you're going to study about how genes and nutrient interacts, and when you learn about it, it's called as nutritional genomics. And when you talk about the transcriptome, so it is all the sum total of all the mRNA molecules expressed from the genes of an organism, which is called as a transcriptome. And then the proteome is the complete set of proteins, which is expressed by the genome. And metabolome, I said, set of small metabolites in the body fluids of an organism, which collectively is called as a metabolome. And when you study about how nutrients are affecting each of these factors, it becomes nutritional omics together. And you also have epigenome, that is how environmental factors have an impact on the gene expression without affecting the DNA sequence. So far, the genomics, transcriptomics, proteomics, metabolomics, It all talks about the changes in the gene structure or function, whereas epigenomics is quite distinct, where we talk about the chemical modification happening in the DNA without changing its sequence. So if you look at these nutrients, for example, vitamin C, we talk about vitamin C and improving on building immunity, but in precisely what happens about vitamin C inside the cells is it regulates the expression of the genes, which actually codes for the extracellular matrix protein. That's again, it's a transcription factor. And it helps in the peroxidation. It helps in the prevention of peroxidation of the lipids and the DNA. So this is how it is actually helping us out in improving the immunity. So it actually, it involves or it interacts with the transcription factor, and thereby it regulates the prevention of DNA and lipid oxidation. And that's why it is kind of involved in immunity. So if we look at each of these nutrients, we are looking at how these nutrients affects the gene expression. So that is nutrigenomics. On the other hand, if we look at how the variation in the genetic factors affects the response to food, it's called as nutrigenetics. So as a whole, we are looking at the gene-food interaction and how each other influences one another is what nutritional genomics all about. So basically, we are trying to speak about the genetic variability among humans, how genes are, the genetic makeup is different between human beings, and how it influences our response to food is what today's session about, nutrigenetics. And as I said, there are two different omics approaches, nutrigenetics and nutrigenomics. It's like two sides of the coin, it's inseparable. And it is targeted nutrition approach. So we are not looking at just the phenotypic assessment, but we are also looking at a single element genomics here. That is why it's called as personalized or targeted nutritional approach. So when you talk about the vitamin C and how it affects the DNA, how it prevents the DNA oxidation, we are talking here about how food is affecting the gene expression. That refers to as nutrigenomics. Whereas here, again, when you talk about the genetic variability, just as I told you, how each person looks different, why we are not identical, why we are all different is because of the genetic variability. And if you learn about how the variability is influencing the food, the response to food, it's called as nutrigenetics. And we don't stop just with learning about the interaction, but we also learn about how these interaction affects and have an impact on the disease outcome. So to understand it kind of easily, it's a simple analogy. Just remember a genome to be a book of life. And then you have the chromosome, 23 pairs as the chapters, and DNA are the pages. And DNA are basically made of the genes. So that's why I said segment of genes are called as words. And nucleotides are like A, T, G, C. These are nucleotides. And spelling mistakes are the genetic variation. So you have a human body, and the human body is made up of different organ systems. When you look inside the organ system, you'll find trillions of cells in your body. And when you look deep down into the cell, you have the nucleus. And when you delve into the nucleus, you will have this chromosome 23 pairs. And when you untangle the chromosome, you will find the DNA. And segments of DNA are genes. And you will have these base pairs, adenine, guanine, cytosine, and thymine, which forms the DNA. And this is what we are more particularly interested about in nutrigenetics, how these are going to be differently arranged in different people and how that difference is contributing to the variability among individuals is what we are more concerned about. So when you talk about the spelling mistakes, so single nucleotide variant or polymorphism, it is about the arrangement of the DNA sequence where one nucleotide is replaced with another nucleotide in this particular individual. And if it is going to be one replaced with another nucleotide or one replaced with another nucleotide, it's called as single nucleotide polymorphism or single nucleotide variant. Whereas if one is replaced with many, it's called as insertion, and one is replaced by none, it's called as deletion polymorphisms. So you have all these different differences in the genes that we carry. It could be one nucleotide replaced with another. So think like our DNA each of our DNA is arranged like this: guanine, adenine, cytosine, and thymine, and it goes on. And if one is replaced, so here if you look at, if the adenine is replaced with guanine in you, so imagine like this is my DNA sequence and that is yours. So here I carry adenine here in this position of the chromosome in this DNA sequence, whereas you carry guanine. And these are the differences in the arrangement which causes our differences, why we look different, why we act differently, and why we respond to food differently. It is basically because of these kind of differences in the arrangement. So if you look here in this picture, father can carry adenine and the mother can carry guanine, and the child can carry either of these. And this child is carrying A adenine in this particular position. So this is what we inherit. It could be either A or G, either from your father or your mother. And this is what makes it so unique and gets tied to the genetic inheritance material. So through personalized nutrition approach, what we are trying to answer is we are trying to answer why two individuals respond to the same diet in a different manner. And this is why we as a nutritionist or dietitian need to learn about genetics, because this helps us to understand the differences in the response that exists with a particular individual to a particular diet. So before going on to the genetic examples like the gene and nutrient interaction, I also wanted you to understand about the evolution of humans and food systems. So as I said, it's always the genes interacts with the environment. Gene doesn't function in isolation. Just because you carry a gene doesn't matter if you're going to have like that particular disease. It's always the nature versus nurture story. So nurture is the environment which nurtures you, and the gene is the nature component that you're born with. So it is always about the interaction between nature and the nurture. So if you look at the environmental changes, humans used to be hunter-gatherers initially, and they used to hunt for food, run for food. They expend a lot of energy and they eat whatever it was available. So they didn't grow anything. I mean, it was like whatever was available, they didn't choose it. So if they have a good meal from the hunt, they eat it, or else they're going to fast. So that's how the initial concept of intermittent fasting came into role, because that was like fasting and feasting. So if they hunt for a big meat, so if they got a big animal, then they feast on it. And for some days, if they don't hunt or if they don't find a good meat, then they just fast. So this is how hunter-gatherers used to live. And there was no cooking, no processing involved in the food. But then slowly they began to settle, humans began to settle in the places of civilization near water resources. And you could see that the hunter-gatherers, when they turned into pastoralists, they started growing their own crops of choice, or crops which were adaptable for that particular climatic condition. So from what was available to what they chose to cultivate, became a kind of a very big transition. So you're not eating what was available, but you rather chose to grow whichever you liked it or you preferred it, or it is adaptive to that particular environment. And slowly after the discovery of fire, cooking started and the processing started. And today we are eating almost like 200% ultra processed food. So it's all like totally processed without any fiber, high in salt, high in sugar, high in kind of dairy. So it is, the food habits have changed and the food processing have changed. So all these changes in the environment have also led to the genetic adaptation in humans. So you could see there's a vast difference that existed, and also how humans and food system evolved. So you could just think about the changes that happened in the environment, and thereby how the humans got adapted to these changes is what determined the genetic variability. Likewise, you could also see, we all know humans originated from African region, and they slowly migrated to different regions in the world. And when they migrated, they also have got adapted to the various food availability, whatever was available And it is also about what kind of weather condition they get adapted to. All these shape the human genome. That's why you could see the difference today between Indians, between Asians, as well as Caucasians and Americans. So no one looks similar, right? We're all humans. We are supposed to look similar. But if you look at the body patterns, or even the BCA of different ethnicities, it's different because of these differences that actually happened many, many billions of years ago. So we need to understand the genes doesn't change overnight. It takes about like a lot of years, decades for these changes to get evolved. So you remember the chimpanzees, we come from the chimpanzee family, right? So it didn't take like just 10 years or 20 years. The evolution happened over billions of years, and then the primates became humans. So it is like, it takes a lot of years, and these were the quite significant changes with respect to food that has happened before. And I'm not talking about rocket science, and this concept of nutritional genomics is not something new. Even if you take a cross book from the oldest library, you could find these conditions written there. PKU is something we all know. PKU is treated with low phenylalanine foods or restricted phenylalanine diet. So PKU is a condition. Phenylketonuria is a condition where there is an elevated level of phenylalanine in the blood, and that is because of a defective gene called as phenylalanine hydrolase gene. And when you have this defective enzyme, the individuals usually have elevated levels of phenylalanine in the blood. And when they have lower phenylalanine containing foods, it automatically gets reversed. So this shows that there is a constant gene-nutrient interaction that exists. So once you make some changes, appropriate changes in the food, you see the difference in the condition. So this is a very typical example of gene-nutrient interaction. But it's not as simple as that when it comes to a polygenic condition like vitamin D deficiency. So if you look here, you know about vitamin D2 and D3 forms. There are two forms, and once it gets into the body, it follows the same biochemical route, and it gets hydrolyzed into calcidiol first in the liver, and then it gets further hydrolyzed to calcitriol in the kidneys. And you could see the red ones are the enzymes, and the enzymes are again encoded by the genes. So the calcitriol, what happens is that it gets into the vitamin D circulation and via the vitamin D receptors, and it gets into the cell. If you see this biochemical pathway, there are so many genes involved here. So there could be like defects happening at different stages of the vitamin D metabolism, and also because of defective enzymes in different genes. So here we cannot say that you take a particular nutrient, then it gets reversed. So you have to understand where the defect is happening, and the genetic testing helps you to determine the effect of these genes and where it is the defect, where or which genes is defective, and thereby you could you can identify the root cause of the deficiency, and then you can actually address which foods or which supplements can actually help you to kind of manage that particular defective enzyme. So whenever it is polygenic, not only vitamin D deficiency, it could be diabetes, it could be hypertension or hypercholesterolemia, or it could be cancer. So these are all multifactorial. Phenotypically also there are a lot of factors involved, as well as there are multiple genes involved in vitamin D metabolism. So the challenge for a dietitian or a nutritionist professional is to understand the biochemical pathways when it comes to polygenic conditions where there are several genes involved. And we also need to find out how are we going to address these conditions at a root cause level, and how are we going to kind of give the right solution for the person. So with this example, this is a very interesting example. So we often get questioned about which is the good type of cooking oil to be used. So when it comes to fat, there are so many dietary dilemmas that exist. So take egg yolks or like fats like margarine or butter or ghee. There's always a kind of controversial statements, whether it's good or bad. But we always take the stand of either say, go to research evidences and then like refer to that, or we give a kind of personalized, biased statement of whatever we have learned with our knowledge gathered, right? So, for example, if someone asks you about which cooking oil is good for health, so we often say the oil which is used in the region or the oil which we know of from our knowledge that we have gathered over years of practice. Or we could also tell them to refer to the scientific literature and then take their own decision about, we are just informing them. All these could be actually kind of a kind of, justified and a kind of, I would say it can be a kind of unbiased decisions, can be evidence-based decisions can be given if we understand a little bit about the genetic makeup. So whatever kind of oil that we suggest, cooking oil, whether it's going to be MUFA-rich or PUFA-rich or it's a saturated fat, any plant-based source, it goes through a series of reaction in our body to get converted into long-chain fatty acid. And these series of interaction actually involves various enzymes called as fatty acid desaturase enzymes. So when we talk about the FADS1, fatty acid desaturase 1 group of enzymes, so it plays a very significant role in the conversion of short-chain plant-based fatty acids to long-chain EPA and AA fatty acids. And it's not only one enzyme it's involved, there are also like series of other enzymes that are involved in these reactions. So if you carry A allele, that is, we obviously term them as excellent converters. They're good at converting the short chain into long chain fatty acids, and poor converters, that is, it is C allele carriers. So they're not so good in converting the short chain fatty acids into long chain fatty acids. So as a result of which, they need some kind of assistance in this conversion. So if you look at the fatty acid desaturase 1 gene and the particular SNP, SNP, we are talking about only this SNP. A allele carriers are good, and whereas C allele carriers are not so good in conversion. So how we can stratify and give recommendation for these two group of people is, so the genotype-based dietary recommendation will be A allele carriers, since they are good at conversion, we are going to provide them with plant-based fatty acids. Any type of plant-based fatty acids, they are going to be good at it. Whereas the C allele carriers, whom we term it as poor converters, were not so good in conversion, we can directly give them animal-based fatty acids, which are rich in EPA and DHA. So this is how you kind of streamline the recommendations or segregate the recommendations based on the genetic makeup. So here the genotype-based recommendations are actually given based on the test results from the nutrigenomic test by analyzing the genetic variations. So you know that FADS1 is linked with the fatty acid metabolism. Then you test for that, and then based on the result, whether you are A allele or C allele, it is actually a test result which gives you whether the person carries A allele or C allele. Then you give the particular recommendation that is appropriate to the genetic makeup. So rather than saying this oil is good and that oil is good, because like each region has its own preferences, but we are not really considering the genetic variation that exists within the human, and this is how the personalized nutrition approach is different from the conventional nutrition approach. And this is a picture about the frequency, the allele allele distribution among different ethnicities globally. You could see that in the Americas, there are more percentage of poor allele converters, because if you look at the ancestral diet, they were really meat eaters, Red Indians who used to rely on meat. Whereas in the Asian countries, it was more of a plant diet. So the percentage of the staples like fruits, tubers, as well as the greens and the plant-based options were more. And as a result, today it has turned into an advantage where they are able to metabolize fats more efficiently than the meat eaters. And the past, we usually ask which oil is best. But from now on, you should ask which oil is good for me, because the oil which is good for you need not be good for the other person. So we cannot actually categorize the foods to be good or bad. It's just good or bad for yourself. So the question and the perception needs to change in nutritional science. And it is also about, it is the it is not about the scientific bias or the personal bias. So we need to think far beyond that. So it shouldn't be answered with our personal choices, nor we should just rely on scientific literature, but it should be correlating all those together for a person, which is good or which is bad. Then it makes the right choice. And likewise, another example where we all can relate to, everybody likes coffee and everybody drinks coffee. So I thought it would be good for you to understand the differences in the genetics and how do we make the recommendations different. So you have different bioactives in coffee: magnesium, aliphatic acids, potassium, melanoidins, polyphenols, diterpenoids, so on. But here we are just looking at caffeine, which is the widely studied bioactive in the coffee. And when you drink coffee, what happens is it mixes in your bloodstream, it reaches the liver, and in the liver it gets metabolized. So usually when we do research in nutrition sciences, we think about one particular nutrient and linear relationship with a disease. So here usually I've seen a lot of studies with CVD, cardiovascular disease risk, how coffee, whether it increases or whether it neutralizes the cardiovascular disease risk. Whereas a nutrigenetic approach, what we look at is we study the interaction between coffee and the genetic makeup, whether a person carries a genetic variation, whereas it's genotype A, B, or C, whether it increases the effect or whether it nullifies the effect or whether it decreases the effect. So this is the basic difference. We don't just look into linearly like whether a nutrient affects the risk of the disease, but we also take into consideration the gene-nutrient interaction and how it impacts the disease risk. So as I said, when you drink a cup of coffee, it gets metabolized with the help of an enzyme called as cytochrome protein 1A2 in the liver, and it gets broken down into simpler metabolites like paraxanthin, dimethyluric acid, methylxanthin, and methyluric acid and methyluracil and so on. So here I just mentioned about CYP1A2, but there are 22 other cytochrome protein enzymes which are involved in caffeine metabolism. So, and this determines the rate at which you break down caffeine in your liver. So if you carry a specific genetic variant in this enzyme, so you actually take a longer time to break down caffeine. Whereas if you carry another type of genetic variant in this particular gene, CYP1A2 enzyme, then you actually break it down at a much faster rate. So this protein or this enzyme determines the rate at which the caffeine gets broken down in the body. And this is what actually makes the difference, whether a person is a slow metabolizer or fast metabolizer. So AA carriers are termed as fast, whereas AC and CC genotype are slow metabolizers. So if you look at the caffeine intake amount, so 1 to 2 cups per day, actually, for slow metabolizer, it does not increase the heart attack risk. And when you increase the dosage, 2 to 3 cups, then it increases the risk of heart attack by 36%, whereas fast metabolizer decreases the heart attack by 22%. So here you can find the difference. So it's not about the coffee or the dosage of the coffee, which is bad. Whereas if the slow metabolizer group category, it increases the risk, whereas the fast metabolizer drinking 2 to 3 cups decreases the risk. So this is how we segregate the person based on the genetic variability, and 4 cups or more per day for individuals less than 50 years of age. I'm sorry, it's actually more than 50 years. It increases the risk of heart attack by 4-folds. So here you could see that it's not the coffee which is good or bad, whereas it is good or bad for the person based on the genes that they carry. Fast metabolizer, they'll be able to metabolize caffeine normally because the enzyme activity seems to be like really fast. It can break down the caffeine, and you can recommend up to 400 milligrams of caffeine per day according to the nutrigenetic principles or the research articles that are referred. And lean and younger people are likely to metabolize caffeine faster than obese and older people. So this is also something of the metabolic type, which actually causes these differences. So what if a slow metabolizer wants to still wants to drink coffee? So they can have grapefruit juice, apples, peppers, red wine, dark cherries, tomatoes, cruciferous vegetables. These are actually inducers of the enzyme. And these actually speed up caffeine metabolism. But still, you can recommend up to 200 milligram of caffeine per day for a slow metabolizer. So even if a person wants to drink caffeine, so you can actually suggest the inducers of an enzyme so that it will likely to speed up. So they won't have like grapefruit juice along with the coffee, but you can actually stretch it during the day. So if they drink coffee at 6, you can give these kind of vegetables and fruits later on after 3 hours so that it will actually metabolize, help them to metabolize efficiently. So here is again a research incept where you could find here the coffee intake and the risk of myocardial infarction is given in the first graph. So if you look at the total population, it's stratified based on the cups, number of cups the person takes every day. So here it's less than 1 cup and 2 to 3 cups, 1 cup per day and 2 to 3 cups and 4 cups. If you look at this side in the y-axis, it's like odds ratio and it's a total population. So here it's about the cups of coffee, and people having more than 4 cups per day are at higher risk for myocardial infarction. And here in the second graph, so it's coffee intake and risk of myocardial infarction among different genotypes. So here it's AA genotype and it is AC and CC genotype. So it's less than 1 cup per day, 2 to 3 cups, and more than 4 cups per day. So here AA genotype, even when they have like more than 4 cups, the risk for myocardial infarction, it's pretty much less. The odds ratio, it's only 1. Whereas the AC and the CC genotype, they are slow metabolizers, and when they have more than 4 cups, only this group of people are at higher risk for myocardial infarction if they have more than 4 cups. So it's not about just looking at the total population and then like segregating them on the cups or dosage of a particular nutrient and saying that people having 4 cups are at higher risk. So it's actually segregating based on the genotype and then looking at how they respond. So you can see there is a difference in the response to a particular nutrient called caffeine in the coffee. So here you could understand that there is a gene-nutrient interaction, and the gene-nutrient interaction differs based on the genetic variability. So it is not the 4 cups which increases risk of myocardial infarction, but it is based on the genotype that they carry, which increases their risk. And it is not only with myocardial infarction, it's also with hypertension. So here again, abstainers, people who don't take coffee, 1 to 3 cups per day, and it is more than 4 cups per day. And if you look at the AA genotype, actually the risk declines when they have more cups of coffee per day. And here the slow metabolizers who are AC and the CC genotype carriers are at higher risk when they consume more than 4 cups per day. So this shows even the 4 cups of coffee or caffeine per day can be benefited for some, whereas it's harmful for others. So that's why I would say it's the food that is not good or bad, whereas the response to the food is going to be good or bad based on the genes that the person carries. So here, by this example, I think it would be very clear that there is a gene-nutrient interaction that is constant, and that's going to determine the overall health outcome. So I hope I make some justice to make you understand about gene-nutrient interaction and how the dots are connected between the genes and nutrients. So I would like to conclude that genetics can be used as a tool in dietary assessment in addition to the existing ABCDs of dietary assessment in assessing the nutritional status of a person. And this is just a snapshot from the practical perspective. I want to give you some perspective to it. So if you look at this, it's taken from a genetic report. So whatever we spoke about, the gene will be mentioned here, the RSID and the genotype, and the scientific strength is also mentioned. So here it is all direct to consumer testing, which means that it is sold directly to consumer without any prescription model. So whoever wants to take these tests can go log in to any website and they can just type in and then make the payment. They get the test ordered and it comes home. So you don't even have to walk outside your house. It's all done in your e-commerce platform. And once you send back the samples, you'll get the reports in the email. So it's simple and very kind of organized. You don't have to go anywhere, just a click and you'll get all these tests done, nutrigenomic testing. And this is what a report contains. It will have a gene name, the RSID, the genotype. So based on this, only a dietitian gives recommendation, whether the person carries GT or CC, and that is what makes you very unique. So if you look at the another example, the FADS1 gene that I spoke to you about, here the RSID is the SNP that they analyzed is 174547, and the genotype for this person is TT. So once you see TT genotype, you also see what it infers to. People with your genotype are found to have typical blood levels of important omega-6 fat and an important omega-3 fat. Okay, this person has to optimize from the diet. So how this testing is done? So you collect the sample, DNA sample, and transport it to the lab, and the DNA gets extracted. Then there is a process called genotyping, and then the results and interpretation is done, and the nutritionist does the counseling with the report that is generated. So where do we get the DNA from? It could be from the cheek swab, the cells under your cheeks, and that could be taken from a swab, saliva test, that is you just spit your saliva, and it could also be obtained from the blood sample. So in all these samples, you have your DNA, and you first extract the DNA from whichever sample that you're drawing, and we go to a genotyping technique called as next generation sequencing technique. And there are different techniques that are available, exome sequencing or whole genome sequencing. It all depends on the cost that is affordable and what are the variants that needs to be tested. Once it's analyzed, you get a result like this, where I showed a snapshot of the report. And then it's basically about understanding the human metabolic individuality and the biochemical pathways. And then you give kind of recommendations based on the biochemical pathways and where the gene is actually involved. So you actually learn about a gene and understand the biochemical pathway, come up with these kind of recommendations based on the genetic variants, and then you counsel a patient using this information. So usually what happens is that we have a blood test. We know about the signs and symptoms of the deficiency. And we also have a dietary assessment, which is done by 24-hour diet recall or a food frequency questionnaire. Then the genetic information is about the genes that are involved. So when it comes to blood tests, the serum homocysteine levels, so if you want to take a kind of vitamin B12 deficiency or iron deficiency anemia or folic acid related anemia, deficiency anemia. So you get a blood test done, you actually ask them for the markers that are related to that, and then watch out for signs and symptoms, dietary intake. And then you also actually check for the genes that are involved in that particular pathway. It's as simple as that. We are already doing these three. Now it's time to actually include the genetic information. So once you do that, it will be ABCDs, and then it will be like this, integrating genetic information and dietary assessment. So you have BMI, waist circumference, weight, height, waist hip ratio, BC analysis. So you also have certain genes that are involved in these parameters and the biomedical parameters as well. So there are genes that determines the HbA1c or the blood sugar regulation. And the dietary assessment, we usually do a 24-hour recall method and food frequency. So when it comes to MUFA, you would know adipo-Q gene is involved, FUFA, SREB1, and FTO, and saturated fat. So you could actually analyze. So you will get familiar with these genes once you start counseling patients. But I'm just mentioning that once you segregate it based on the parameters that we usually check, this is how you integrate the genetic information in the dietary assessment. So I hope precision nutrition approach is a bit clear and it would have invoked some interest in you to explore more. So we are not going to look into the plate and say you eat based on the activity levels or based on the dietary habits, but we are also going to start looking at nutrigenomics, microbiota, metabolomics in the deep future. And I hope this will make nutrition science more and more prominent in healthcare, and it would become a central element in healthcare rather than focusing. And the intention of personalized nutritionist approach is more towards prevention is better than cure. So preventive nutrition is going to take a central stage when it comes to longevity, when it comes to aging. And that is why we say precision nutrition is the way forward. And it is all about combined genotyping and phenotyping in nutritional assessment. Hope things are clear. And I would like to wrap up saying a big thanks for the opportunity given to present about precision nutrition today. And I would also like you to invite to come and join us on the 31st of January and 1st of February for an international conference happening in Coimbatore, which is on precision nutrition. And you could check also www.nutriome.org. So this is something that we're doing as an initiative to spread awareness on precision nutrition. So if you want to learn about precision nutrition, you could definitely join us. Thank you again.
Thank you. Thank you so much, ma'am. I extend my sincere appreciation to Dr. Janani ma'am for her lucid and insightful presentation. So, ma'am, can we take some questions?
Sure.
Ma'am, this question is from Dr. Jyoti Pratisya, is from West Bengal. Kindly highlight about the integration of gut microbiome and nutrigenomic data, developing personalized dietary interventions and disease prevention. Just one, two lines. These are huge things, definitely. Ma'am, can you please elaborate in one, two lines? You have explained very well in depth, but in a gist.
Can you just come again with a question? So she wants to learn how we can combine gut microbiome and
Yes,
nutrigenomic data, is it?
absolutely, ma'am.
Okay, see, there are different principles. There's no gold standard till now. So how we actually combine gut microbiome and nutrigenetic approach is gut microbiome still needs a bit of kind of in-depth interpretation, because it's so vast and people are trying to just figure it out how we can actually connect it with a particular disease. So all they give recommendation is what foods to include and what foods to avoid when you look at a gut microbiome report. Then when it comes to genetic variation analysis of SNPs, so you could find few SNPs in the nutrigenetic report. And then we also look into what kind of recommendations can go in. As I said, the companies who are into nutrigenetic testing doesn't give recommendations for each SNP. So it's usually a generic recommendation based on the nutrition recommendations that they give. But as a nutritionist, when we combine both information, so it's going to be understanding about genetic variants and also the gut microbiome to a certain extent. You could give what are the foods to include and what are the foods to avoid, and what are the kind of strains that has to be included. So if it's going to be prebiotics, what kind of prebiotics have to be included for that particular condition, if there is any particular condition needs to be targeted. But as such, there is no protocol that exists. And still, it's to a certain extent, it's kind of a naive information that you get from the gut microbiome report. So if you look at any gut microbiome report, the end result or what information you could apply is you will know about the bacteria present, the diversity and the abundance, as well as the end of it will come to what foods to include and what foods to avoid. Whereas when it comes to genetic variation, to a certain extent, you could predict about the percentage of macros and the micros, as well as there are sensitivities that are usually tested. So it is about including and make it more precise. But as I said, there's no gold standard you could just play around with, but you are just adding some elements to the nutrition plate that you have already made up from the ABCDs. So these are going to just add few elements that could make it more precise.
Absolutely, ma'am. Very nice. I nicely explained, ma'am. I think Jyoti have his answer, and we can take another question, ma'am, if it is possible. That Ms.
Sure.
Aishwarya Das has asked, "Good evening, ma'am. Is there any impact of genetic profile on HbA1c level, as fasting and PP is coming normal, but A1c is high? Kindly highlight." Can you please elaborate in one line about
Yes.
HbA1c level?
Yeah, it's a good question, because you see that over an average is always like it's not coming down. And this is what they say personalized nutrition or precision nutrition can help, because there are also some people who actually can eat ice cream and still the spikes are going to be less. So this means that the ice cream is not actually the reason for your blood sugar spikes. So by understanding the analysis, like analyzing their gut microbiome or continuously monitoring their blood glucose levels along with the gut microbiome and nutrigenetic test, can help them to figure out where they are missing, or where we can identify what is actually causing the blood sugars not to be regulated. As you said, only one parameter, which is quite an average and a predictor of blood glucose, is not getting managed. So this could be actually kind of intervened with all these tests that I told you. There's no specific genetic variations or specific things in gut microbiome that you could check for HbA1c, but you could actually figure out the in total about the genes that are regulating the blood glucose. We can find it out and see if there is any correlation that exists. So there's no one-line answer for this, but we need to really check what are the genes that are involved in blood sugar regulation.
Absolutely. Thank you so much, ma'am, for this insightful answer. I think Miss Aishwarya have this answer. And one more question we can ask, ma'am, from Mr. Ansel Thomas. Good evening. Thank you, ma'am, for this session. It was highly thought-provoking, informative. The routine practice, how to integrate personalized nutrition in disease like CKD or fatty liver disease, where the routine assessment is heavily reled and biomedical and other clinical parameters. So, also, ma'am, how much do you have the phenotypic precision nutrition? Is it possible? Help.
Yes,
So,
that's a good question. When it comes to diseases, it's still an area needs to be kind of, we need to look upon, because in hospital settings, no one has kind of included these tests as as a part of the protocol or the routine test. And I would like to make two things clear. The nutrigenetic test is done only once. It's just like your blood type. So once you find it out, you don't have to keep repeating it. You know that you carry this genetic variation that's going to be there for lifetime. Only the phenotype can be changed. Whereas the gut microbiome is quite dynamic. It keeps changing. And that you can go in for routine test. So it's going to change as you change your diet. So it's very dynamic in nature, and it's quite reversible as well to a certain extent. So what we can do is nutrigenetic test can be done once, and then those results can be taken, and then we can interpret it and use those interpretations and keep seeing how the phenotype gets changed. Whereas the gut microbiome test, usually the companies who are testing right now also suggest them like once in 3 months or once in 6 months. Again, the cost is one of the important factors. So people can't afford it like every 3 months, means you can suggest them to get for 6 months, because there you can see the differences. The progress can be monitored. Whereas nutrigenetics is just one-time test. You can't keep seeing the changes as you see it with other parameters.
absolutely, ma'am. Actually, the number of tests are very less in India. And, ma'am, one more question from my side. Basically, should future public dietary guideline consider genetic differences instead of one common recommended for all diet plan? Like, is there any plan of setting up any goal of public dietary guidelines? It will be helpful for the common man in India. So, is there any plan, any suggestion from the senior dietitians or anything? It will be helpful for the common people.
Yeah, I think you would have all seen like last week, from the Reliance, they said like 1,000 genome testing. I'm not relevant to your, whether it's going to be relevant to the guidelines that you mentioned. But when it comes like 1,000 rupees, it will become like a geo. Everyone will have a geo SIM. So everyone is going to get this genome tested. Then that means that every nutritionist will have a nutrigenetic report ready. Then it is possible to come up with guidelines of how to integrate or how to interpret. Unless and until we can't have guidelines as such, like for a public forum or a public, kind of use. But definitely when it is kind of normalized, then it is very much possible that we come up with these kind of guidelines. And it's also about the accuracy of these tests. So those are actually getting more kind of better with time. They are getting accredited with various licenses. I could say that accuracy of the test is getting improved. But apart from that, we can't publish like guidelines since this is still unregulated in Indian market.
Uh, relevance. Yes. Thank you once again, ma'am. I'm extending my sincere appreciation to Dr. Janani, ma'am, for her lucid and insightful presentation. Her expert integration of precision nutrition and community health have provided us with a valuable scientific perspective, right? And practical relevance. I also thank IIP in India Association and the Community Nutrition Core Group, all the members, Ranu ma'am, the founder member, the chief program officer, Shomandu sir, and other program officers, and all the executive bodies, but the president of IIP in India for organizing such a insightful, meaningful academic platform and participation of this attentive and active involvement. I am more confident that the knowledge shared today will strengthen our future research and clinical practice and community nutrition initiative. Thank you everyone for your valuable participation, and thank you ma'am for your active participation, and your presence means a lot to us.
Thank you.
We hope we can organize such a seminar once again, webinar, and definitely, ma'am, you will be our guest of honor.
Sure, sure.
I
Thank
have
you.
shared
Thank you,
ma'am's
ma'am. Thank you,
email.
sir.
I have shared ma'am's email ID. So if you have any queries, you can send a mail to ma'am.
If
It is
yes,
really insightful. Thank you, Souvik sir, for inviting ma'am and acknowledging her. We get to know many things. These are the simple things, everyday life, that which oil is good for whom and why this oil and why coffee is bad. You have made the thing very simplified. Thank you. Thank you, Janani, once again. So we'll meet you soon. Okay. Bye bye. Good night.
thank you.